Remember that myofibers are full of those contractile organelles called myofibrils. And myofibrils, those little rods inside of the big myofiber, those guys are made of small little protein filaments called myofilaments. Some are thicker, they're made of myosin, they have a little thicker consistency, and the actin filaments are a little thinner. And because the myosin and the actin filaments line up in these alternating rows, it creates these striations on the surface of the cells. So we get those dark A bands next to the light I bands, just alternating A band, I band, A band, I band, the dark light, peak staining that we're seeing. So today, we're going to start talking a little bit more about muscle contraction. In order to understand how muscles contract, we kind of need to bring in a neuron. We talked about how our skeletal muscle is voluntary tissue. That means you have conscious control over when these cells are contracting. Well, that conscious control comes by the way of your nervous system. So these neurons here that are coming out of the spinal cord, they're going to go ahead and control or communicate with these myofibers, with these cells. Those neurons are called motor neurons. So we're going to tie in the nervous system just a tiny bit today with a little bit of an introduction. And it's actually kind of perfect timing because this unit continues with the nervous system on Thursday. So we will continue with the nervous system beyond this. So this is like a little taste of the nervous system blended with the muscular system. So we need to understand neurons first. So the shape of a neuron is pretty unique. It's a really unique looking cell. Neurons have a nucleus, right? They have a plasma membrane. They have cytoplasm with all kinds of organelles inside. But you can see here that neurons have this really unique shape of having kind of little extensions branching off of the main part of the cell, I should say. So that main part of the cell here is what we call the cell body. And the shorter extensions coming off are called dendrites. The dendrites in the cell body are collectively what we would describe as the receiving end of a neuron. So neurons only communicate in one direction. It's not a back and forth, right? It's one direction only, like a one-way street. So there's got to be some signal coming into the neuron, and then there's going to be a signal going out of the neuron, right? And it's always in one direction. So we say the inside, the receiving side of the neuron is over here. It's the dendrites and the cell body side. What leads away from the cell body, right? The outward kind of signal going out is going to get carried by a longer extension called an axon. So here's the axon of this neuron, and at the end of the axon, it's really common for the axon to branch quite extensively. In fact, an axon could even split and branch even earlier than this. But the key about an axon, no matter how many branches there are or where those branches take place, the axon is always the signaling side of a neuron. So signals, again, always travel in only one direction in our neurons. They come in through the dendrites and the cell body, and then that neuron, if it's going to communicate, will send a signal through its axon to the ends. The ends of the axons are called the axon termini. One of them would be a terminal. You'll see various terms for this. Sometimes it's referred to as like a terminal bouton or axon terminus. Same thing. It's all the same thing. The ends of the axon. Okay. The other thing that we have in this illustration is these cells, these yellow cells that are wrapped around the axon. These yellow cells are called Schwann cells. A Schwann cell is a really interesting cell of our nervous system. It insulates just a small section of an axon by wrapping around the axon in that small section, wraps around the axon about 100 times. So it's like a tube of toothpaste, wrap, right? All the cytoplasm of the Schwann cell kind of gets squeezed to the outermost layer, and what's really wrapped around the axon there are like 99 layers of plasma membrane, right? It's just layer after layer after layer. And that plasma membrane, is made of phospholipids primarily. And so all those phospholipids are providing an insulation to that section of the axon. section that's wrapped by a Schwann cell. What this does is it speeds up the signal. So if and when this neuron signals, right, the signal is going to start at the beginning of the axon. It's going to travel down the axon in that one direction. But instead of the signal having to hit every little point along the axon, it's able to skip to these exposed gaps between the insulated regions. The exposed gap is called a node of RANVA. So the signal will start at the beginning of the axon and then skip to the first node of RANVA, and then to the next node, and then to the next node. And so it's able to jump these kind of insulated regions of the axon. It's said that having a myelinated axon, having all of these Schwann cells providing, we call it a myelin sheath, the signals travel about 1,000 times faster down those axons. So it really does a good job of insulating, and really the end effect is to speed up the signal. So a little breakdown here. The axon, again, is the signaling site of a neuron. This signal is an electrical signal. We're going to see that it involves the movement of ions, positively charged ions. And this change in charge that happens along the axon is called an action potential. So in the nervous system, we don't say the neuron signals, we don't say the neuron fires, we say that there's an action potential. Right? That action potential is the signal. It is the electrical event. And then, as I mentioned, all of those Schwann cells that are covering the axon collectively create this thing we call a myelin sheath. Right? So all of these yellow cells together would collectively make up the myelin sheath. And that is that this axon has all this insulation with tiny open gaps. The tiny open gaps of exposure are the nodes of RANVA. The termini, the axon termini or terminals, are at the very ends of an axon. Down here at the terminals, we're going to see this is where this neuron stores the chemical signal that it's going to release. So the thing about neurons is they don't actually physically touch their target cells. We're going to see in a minute here that a neuron, called a motor neuron, with its dendrites and cell body, here's my axon, I'm going to need a bigger axon, add some stuff to this. There we go. Right, there's its axon. I'm going to make this axon really long here. And we're going to go ahead and give ourselves a little axon terminus down here. And maybe there's another branch. Axon termini branch all over the place. Yeah, but this is going to be my neuron. That neuron is going to signal to a muscle cell, a myofiber. But this myofiber and this neuron never touch one another. So if this is my myofiber's sarcolemma, remember myofiber has that cylindrical shape? This is the sarcolemma of the myofiber. And there's the plasma membrane of the neuron. Right? Here's its axon, there's the axon terminus. Right? Those two plasma membranes never touch. So what's going on down here? How does this muscle cell receive the electrical signal? Right? It's not conducted from cell to cell. It's not like the electricity jumps. Right? This electrical event or this action potential doesn't jump, it can't cross space like this. Right? You don't get electrocuted just by being close to an outlet. You actually have to like stick your finger in the socket. You don't, please don't. Right? But I mean, that's the thing, right? So electricity doesn't jump through space. There's nothing there connecting these cells. So how does this muscle cell know that it's time to contract? Know that the neuron just fired an action potential? The answer is what's happening down here. At the axon terminus, all neurons, no matter what kind they are, they're always going to store a chemical called a neurotransmitter in small membrane-bound bubbles called vesicles. So those are my vesicles, and inside of each vesicle are tiny little chemicals called neurotransmitters. You've probably heard of some: dopamine, glutamate. Well, the neurotransmitter that's stored here in this motor neuron, and it's a motor neuron because it's controlling a muscle cell. So this motor neuron will always release the same neurotransmitter. Muscle cells respond to a neurotransmitter called acetylcholine. I'm just going to abbreviate it ACH, and that's fine for you to just abbreviate it as well. But it's called acetylcholine. So acetylcholine ends up getting exocytosed into this little space here. The muscle cell has an acetylcholine receptor in its membrane, and when the acetylcholine sticks to the receptor, that's going to be the trigger that the muscle cell knows, oh, I've just been told it's time to fire, right? It's time to contract. So that's where we're headed, is getting to this place where acetylcholine gets released. But in order to understand that, we kind of have to go through the whole thing of this action potential firing first. So the big job of the axon terminals are to store the neurotransmitters, right? This is where the signaling takes place. This is where the magic happens. So there are two things that have to happen here. In order for a neuron to communicate, number one, it has to become electrically excited, right? This is what we call the action potential. The action potential is the electrical signal that travels down the axon, right? We'll talk about this. When the action potential reaches the axon terminus, it triggers what we call synaptic transmission. This word comes from the term for this connection. This little gap between the two cells is called a synaptic cleft. And it's just full of ground substance, right? It's just a little fluid outside of the cells. Remember, what's outside of each individual myofiber is endomysium. Right, so we would be in the endomysium area here. It's areolar connective tissue. It's kind of loose material. It's got a lot of space for ground substance. So our synaptic cleft is a lot of fluid. The connection between a neuron and any cell it's communicating with is usually referred to as a synapse, just generically. A neuron communicates at a synapse. So this whole area would be called the synapse. Specifically, the synapse between a motor neuron and a muscle cell gets a special name. It's called a neuromuscular junction, NMJ. So this whole thing, we'll go here, a little bit bigger. That is the neuromuscular junction. So the synaptic cleft is the space that's found within that neuromuscular junction. These are terms that you'll see on some histology slides. We'll get there in our nervous system. We'll see this neuromuscular junction. I have a model of it. All right. We gotta build our neuron just a little bit. So we talked about how these dendrites are the shorter extensions. I'll add a nucleus here in the cell body. We need to add some new terms here. So the beginning of an axon, this beginning location is called the axon hillock. And I'll go ahead and add some Schwann cells that region. And I'm gonna leave some decent sized nodes of RANVA. You get the idea. I'm just gonna box it up for the rest of them, but be all my Schwann cells, and all the gaps of open space, open axon, are the nodes of RANVA. So I'm gonna go ahead and do a little labeling. So Schwann cell. All together, all of the Schwann cells make up the myelin sheath. I'm not gonna add that to my label because it would get a little crazy. Open bits of the axon. It's called a node of RANVA. All those little nodes. And then of course our axon terminus at the end, so we'll label that as well. Before I get into any of that, we have to talk now about membrane proteins. So we're getting into like a little bit of molecular anatomy right now. So we're going to talk about some membrane proteins that are related to what we actually started talking about weeks ago in the cell lecture. We talked about how some molecules get across a plasma membrane by simple diffusion, right? Oxygen, carbon dioxide, those guys can easily diffuse through phospholipids. But anything that has a charge or is too large cannot. Okay? But one way those charged or large molecules can get across, is through facilitated diffusion. And that doesn't require any energy, as opposed to like active transport that does require energy. So we're going to be actually talking about both of those with our neurons and with our myofibers. We're going to talk about active transport, and we're going to talk about facilitated diffusion. So first, let's talk about active transport. I'm actually going to pause this whole show for a moment. Come over here for a second. So active transport, the one that was the example that was given in that lecture, the cell lecture, is the example of the sodium-potassium pump. So sodium is a positively charged ion, it's Na+, and potassium is a positively charged ion, it's K+. That's sodium and that's potassium. And this sodium-potassium pump is an active transport protein that's found along the plasma membrane of both of these cells. They're loaded with sodium-potassium pumps. So I'm going to use a little like symbol here. We'll just call it a little circle. That's going to be my sodium-potassium pump symbol. And I'm just going to show that they're everywhere. Right? These sodium-potassium pumps are everywhere, all along both cells. So what are they doing? The sodium-potassium pumps, all of them, are always working along both of the cells to always pump 3 sodiums out of the cell and 2 potassiums in. 3 sodiums out and 2 potassiums in. Everywhere. All along the cells. Can you see that this creates a really high concentration of sodium outside of our cells and a pretty low concentration of sodium inside? Yeah? So we get this, we call it a concentration gradient. When there's a difference in concentration in one location versus another, it's called a gradient. So sodium concentration becomes really high outside of the cells. I'm just going to put a big Na. Lots of sodium outside of our cells, everywhere. Lots of it. And pretty low levels of sodium inside of our cells. The other thing that happens then is by pumping all this potassium in, is we create a high concentration of potassium inside of our cells and a pretty low concentration of potassium outside. Okay, so all this potassium ends up in the myofiber, all the potassium ends up in the neuron. All over, lots of potassium. Sodium outside, potassium inside. So far, so good. So if I'm going to list what the sodium-potassium pump does for my cells, I'm going to say number one, it creates a high concentration of sodium. So I'm going to put a bracket around sodium. That's kind of a shorthand way of describing concentration. It's a high concentration of sodium outside of the cells. Number two, it does the same thing for potassium, but inside, different location. So a high concentration of potassium inside of the cells, higher than outside. You could list this as, creates a concentration gradient for sodium, creates a concentration gradient for potassium, however you want to describe it. But this is pretty simple, makes sense to me. It's easy for me to remember. So that's where I'm starting. Right? But there's one more thing that my sodium-potassium pump does. Notice that this pumping of these positively charged ions is uneven. Right? And it's always going to be at this 3-to-2 ratio. Always 3 sodiums out, 2 potassiums in. And then 3 sodiums out, 2 potassiums in. If there's an excess of sodium, it's not like it keeps pumping sodium out without ever pumping potassium. It takes turns. It's the way the pump works. So we always are going to get 3 sodiums pumped out, and then 2 potassiums pumped in. And then 3 sodiums pumped out, and then 2 potassiums pumped in. Always going to be this like one positive charge difference. You think about it. Just ignore the fact that it's sodium and potassium. Right? If we just count the positive charges, there's going to be a difference then in positive charges, and pumping more positive ions out and fewer positive ions into the cell. And that creates a difference in charge. It creates a more positive external environment and a less positive internal environment. Another way to describe something as less positive is to say that it's relatively negative. So we use this term relative because it's not like the inside of the cell is absolutely negative. There's no negative ions involved here. But relative to the outside of the cell, right, it's less positive. Another way of saying less positive is more negative. We use this analogy of like height. Have a really tall person, maybe 6'3", that's pretty tall, right? You're taller than that, clearly, right? But we bring like a 7-footer, like Shaquille O'Neal or somebody in here, and our tall guy looks relatively short, right? Or a tall girl, whoever. So that's the same idea, right? It's not like we have negative ions in here, but compared to the outside of the cell, the inside is relatively negative. And the outside of my cell is more positive, or relatively positive. And that's going to go for both cells because of this pumping, right? Because of this 3-to-2 pumping. So inside of the neuron, it's relatively negative. Outside of the neuron, it's relatively positive. More positive outside, less positive inside. That difference in charge is referred to as a polarity. So the third thing that this thing does is it establishes, we call it the resting potential or the resting polarity. For both of our cells. And I say resting but you know the sodium-potassium pump requires ATP, right? It's a pump protein, it's active transport, and it is working all the time. So even when your neurons are not firing, even when your muscles are not contracting, they're still burning through ATP, right? They're still needing ATP to power these sodium-potassium pumps. At rest just means not firing, but it doesn't mean the cell is doing nothing, right? So this muscle is at rest when it's not contracting. This neuron is at rest when it's not firing in action potential, but they're still very active all the time using the sodium-potassium pump. Okay, one membrane protein down. We've got 3 more to go. No, 4 more to go. Okay, you ready for the next one? Okay, our next membrane protein, it's called a voltage-gated channel. No, let's not go there. Well, we need to get there anyway. I'm going to start actually with a different one. I'm going to start with a channel protein for facilitated diffusion. This channel protein is only found up here. Let's go ahead and draw like a little rectangle. Blue rectangles. This channel protein is only found on the dendrites and the cell body. So that little channel protein is called a ligand-gated channel protein. And this one is a channel protein for sodium. So we're going to make it really specific. It's a ligand-gated sodium channel protein. Maybe somewhere in your notes you want to write down that this is for facilitated diffusion. And if you remember, facilitated diffusion does not require any ATP. So a channel protein is basically like an open door or an open window. Remember that whole idea that if I sprayed like perfume in the room, it's going to end up spreading, diffusing throughout the room. If we opened a door, right, then the perfume can diffuse outside of the room. Right? No energy is going to be required. Just the open door is what lets it out. So that's the same idea here. If this channel protein opens, then sodium can diffuse through. And diffusion occurs only in the direction of that concentration gradient. So these ligand-gated sodium channel proteins have a few characteristics. Number one, they're only found on the dendrites and cell body. Number two, they are triggered by a ligand. So they're normally closed. What opens the channel? What opens the door? A ligand is a chemical signal. It's just a big molecular biology word for a chemical signal. So my motor neuron here, it is all your motor neurons. We're going to learn this in our nervous system. All your motor neurons, cell bodies are like way up in the spinal cord. The axons are stretching out through your body to get to your muscles, wherever those muscles are that they're controlling. This motor neuron is not making the decision. Your spinal cord is not making the decision that it's time to contract a muscle. What makes that decision?
Brain.
Yeah, your brain, right? You decide you want to contract a muscle. So this motor neuron is not the one deciding that it's time to fire. It's being told, right? So there's got to be some brain neuron. A brain neuron is going to signal to this motor neuron. And all neurons kind of have the same anatomy. So what's really happening here is some brain neurons, axon terminus is down here. Right? And remember that axon terminus has these vesicles with neurotransmitters. When this brain neuron fires, right, from the brain, then these vesicles spill their neurotransmitters out onto the receiving end of my motor neuron. The neurotransmitters are the ligand. That's the chemical signal. So this little black neurotransmitter, this little spot, sticks to the sodium, ligand-gated sodium channels, and that's what opens the gate. So if there's a ligand, right, if there's a signal around, chemical, then these little sodium channels are gonna open up. Now we're ready, right? We're set. Sodium diffuses into the cell. Right? And the more of those ligand-gated sodium channels that open, the more sodium diffuses into the cell at the receiving end of the cell. So far, so good. No.
Can you repeat that one more?
Yes. The more of those ligand, right, or neurotransmitter, right, that's the ligand. The more of those ligand-gated sodium channel proteins are sticking to their neurotransmitters, the more of them are open. And if they open, then sodium can just diffuse through them. Because we have a high concentration of sodium outside of the cell and a low concentration inside, the direction of diffusion will be from out to in. So the result is when they open, sodium diffuses into the neuron. Right? That's really the end of the story. Just try to break it down.
So the ligand opens the channel and then sodium floods into the axon.
Dendrites and cell body at this time. Dendrites and cell body. Okay. So we're starting to see some changes in our neuron right now. Okay. Having all this sodium flood into the dendrites and cell body, right? Or trickle in, depending on how many of these things open up. We're going to see a change now. Right? What's going to happen here? My resting polarity, or my resting potential inside of my neuron, you can measure this, right? If I were to take, disregarding what's happening up there at the cell body, if I were to take two little probes, there's something called a voltometer. So voltage is a measurement of charge. If I were to take these two probes and compare the inside to the outside, the inside of the cell is 70 millivolts less positive than the outside for neurons. This is different for muscle cells, but for neurons, 70 millivolts less positive. You don't really need to know these numbers for my class. You'll learn them in physiology. It'll help maybe if you have them in your notes. But the measurement there at rest is negative 70 millivolts. Negative 70 just means that it's 70 millivolts less positive than the outside. If you understand this, then it kind of helps to recognize that, like, when the sodium diffuses in up here, right, at rest, the cell body and the dendrites, that negative internal environment was negative 70 millivolts. But what happens then when sodium starts to diffuse in? Right? It gets more positive, more positive, more positive, more positive. Negative 69, negative 67, negative 65, negative 60, negative 58. Right? We're going to keep getting more positive. for this neuron to fire, we don't want it to fire just like accidentally. So there's a trigger potential. We call it a threshold potential. When this neuron reaches negative 55 millivolts, negative 55 is the threshold that will initiate that action potential. So when enough sodium diffuses in up here at the receiving end, really, when enough of these ligand-gated sodium channels are opened and enough sodium diffuses in, when the cell up here reaches negative 55 millivolts, it's going to signal. It's going to fire its action potential. And it will not at any voltage below that. Negative 56, no way. Negative 57, no way. Right? So there's a little wiggle room here. It's like making sure it's really time to fire. What is so magical about negative 55? So it turns out there are 2 more memory proteins we need to add to our list here. I'm going to draw these like little triangles because they are voltage gated. So we've got one that's a voltage gated, not that marker. There we go. We're going to call this our voltage gated sodium channel protein. Do green for the voltage gated potassium channel protein. Both of those are only found on the axon. The axon terminus, the axon hillock, anything that has to do with the axon. But they are not on the cell body and they are not on the dendrites. So I'm going to add some voltage-gated proteins here. Voltage-gated potassium, and it doesn't matter which direction I'm drawing my arrows or my triangles. Just the triangle is the voltage-gated protein. Voltage-gated potassium, they're at the axon hillock, they're at all of these nodes of RANVA. They're not in the areas covered by Schwann cells because those are insulated by that myelin sheath. These guys are everywhere. Okay, so what does it mean to be voltage gated? Again, you can write somewhere in your notes. This is another protein that's used for facilitated diffusion. Just like my ligand-gated ones, all they have to do is be open, and sodium or potassium can diffuse through them. The question is, when are they open, right? So they're normally closed. Just like the ligand-gated sodium proteins, they're normally closed. The voltage-gated proteins are opened by a particular voltage. You know the voltage, it's that threshold. Negative 55 is the magic number, is the magic voltage. So what will happen? Just add that little note here. So location-wise, those 2 are found on the axon, hillock to terminus. And negative 55 is the trigger to open the gates. Right? So neurotransmitter was the trigger to open this one. The voltage of negative 55 is the trigger to opening those 2. Both of them. So let's come back to what's happening here now. Sodium starting to diffuse into the cell body and the dendrites. The inside of the neuron here is going from -70 to a little bit more positive to a little bit more positive. At some point, it might hit -55. If and when it hits -55, nothing's gonna happen up here. But what's gonna happen is the voltage-gated channel proteins at the axon hillock are gonna open. And I just drew a couple, but this sucker is loaded. The axon hillock and every single node of RNVA are loaded with voltage-gated channel proteins, both sodium and potassium, tons of them. So all of these voltage-gated channel proteins are going to be triggered to open when the cell reaches -55. And that -55 hits the axon hillock first, right? It's not -55 down here yet. It's like the perfume diffusing, right? It's the front tables first. So sodium's coming in, This area gets -55 first. All of those channel proteins are triggered to open. Now the timing is kind of unique. What'll happen, and this is more molecular biology than you need to understand, but what'll happen is the sodium channel proteins open first because of the way they open like a sliding door. Like the channel protein itself has this unique shape and it opens like an elevator door, right? It just slides apart. And so it's like an immediate opening. The voltage-gated potassium channel proteins, they're triggered by the exact same -55 millivolts. So they will open also. However, the way that these guys work is there's like this swinging ball that gets moved out of the way. And that causes a fraction of a second delay. So the sliding door opens first, sodium floods into the cell. The swinging ball is in the process, and eventually it opens, and all this potassium floods out of the cell. So sliding door, swinging ball, doesn't matter. What you need to know is that the sodium channels open first, the potassium channels open second. That was just my explanation for why. Sodium channels opening first, all this sodium that's outside floods in. And now the inside of the cell, right at this location, gets really, really positive. Like positive 50 millivolts, positive 100 millivolts, super positive number. Right? We go from -55 all the way up to positive in like, 0.3 of a second. It's really, really fast. Again, that's more physiology stuff. It's super fast. By the time the slightly delayed potassium channels finally open, the inside of the cell is already super duper positive. So when those potassium channels that were slightly delayed, when they do finally open, potassium that's loaded inside of the cell diffuses out. And by losing all of those positively charged potassium ions, the inside of the cell in that same location returns back to a negative potential. If we were graphing this, it would look something like this. It would look like -70, right? That's at rest. A little bit, and this is time down here on the x-axis. So hanging out at rest for a long period of time. As soon as some brain neuron tells the motor neuron, hey, it's time to fire, the inside of the cell gets a little more positive, a little more positive, a little more positive, through sodium kind of trickling in at the dendrites in the cell body. Eventually, let's say -55 is up here. Eventually, when we hit that -55 millivolt, right, that's the trigger for the action potential. All those voltage-gated sodium channels at the axon hillock open first. The inside of the axon hillock is super duper positive. a fraction of a second later, right, all those potassium channels open, the voltage-gated potassium channels. Potassium floods out of the cell, and the cell returns to a negative potential again. This rise is due to sodium coming in, and this fall is due to potassium coming out. If I was measuring what's happening, the potential, measuring the voltage at the axon hillock, just going over time, just through time, that's what I would see. Right? At rest, starts to get a little more positive when the brain neuron is firing, but it doesn't do anything until it hits negative 55. That's when those voltage-gated sodium channels open, and then those voltage-gated potassium channels open. And this is the consequence of that. The cell gets really positive and really negative. One last comment. This getting really, really positive and really, really negative, this is the action potential. This is it. When I say there's an electrical charge, there's an electrical event, the neuron fires, that's what I'm saying. When you learn more about this in physiology, that's what it is. This action potential is just that. It's getting really, really positive, followed by getting really, really negative. You can maybe then imagine if this is happening at the axon hillock, when all of this sodium floods into the cell, it doesn't just stay there at the hillock, right? All this sodium flooding in at the hillock diffuses. And it's enough, and it's always enough, to bring the next set of voltage-gated channels to negative 55. So it's gonna happen like a domino effect, right? If the first set of voltage-gated channels go off, then that action potential that happens here will trigger it here. And this guy will trigger it here. Then this guy will trigger it here. Every single node of RANVA, those gates will open, right? Boom, 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 one right after another because of this domino effect. Because of one triggering the next, triggering the next, triggering the next. So the entire action potential would be the entire cell experiencing a wave of getting positive followed by a wave of getting negative. Okay, questions? Yes?
Wait, so the ligand gate is what lets the sodium in until it gets to that 55,
Yes.
and then everything else happens.
Yes. Yes.
So it's like all part of one
This
process.
has to happen first. These ligand-gated channel proteins at the receiving end of the neuron, the dendrites in the cell body. When the neurotransmitters around, those guys open. That's what gets enough sodium in. So this right here is all happening because ligand-gated channel proteins at the cell body and dendrites are open.
So then it like equals out negative 70 again after the potassium.
Oh, great question. So again, I was going to leave that for your physiology class. There is a period of time here where the cell can't fire again because yeah, we're back to a negative potential, but our ion positioning is all off at this point. Right? What happened to get back here is potassium flooded out, and sodium is left in the cell. So we gotta reestablish, your sodium potassium pumps have to work hard to return sodium and potassium to their proper locations and get back to maintaining that rest. So this is kind of like a temporary situation. Yeah, it still needs to be corrected.
Is that when fatigue happens with your muscles and stuff like that?
Oh, so fatigue is more related to ATP, ATP supply issues, and that is usually due to glucose and oxygen deficiencies. Yeah. Yeah, we'll get there. still haven't gotten to, like, how do these neurotransmitters get released? Right? We've got an action potential. We've got this, firing, this getting really positive, getting really negative, positive, negative, positive, negative, positive, negative, positive, negative, all the way down, node to node to node to node to node. What happens here? Right? It's not just sodium flooding in and potassium flooding out. We have one more protein to add here, one more membrane protein. This guy is another voltage-gated protein. So I'll draw the little triangle again, but different color because it's for a different ion. So my purple triangles only found at the axon terminus. Those guys are voltage-gated, I'm just gonna put Vg, calcium proteins. Calcium is a doubly positively charged ion. And it's again a channel protein, so it's for facilitating. So this guy is only located on the axon terminus. So again, just like all my others, closed at rest. Doors are closed. So based on its name, I hope you can maybe predict that it's going to be a change in voltage that triggers the opening. Right? The gates open due to a change in voltage. And I'll be honest, I don't know if it's negative 55. I don't know what the threshold is for these guys. But a more positive voltage than negative 70 is what's needed here. Okay, so we know that when that action potential reaches the axon terminus, the inside of the terminus gets really, really positive from all that sodium flooding in. Okay? The potassium channels will then open, and then all that potassium floods out, and the terminus gets really negative again. So we know that that action potential will hit the axon terminus. When it does, the getting positive is what triggers the opening of these voltage-gated calcium channels. So to understand which way calcium is going to diffuse, we need to know the calcium concentration gradient. Your body, your physiology, your homeostasis does a really good job of maintaining pretty high levels of calcium outside of our cells and very low levels of calcium inside of our cells for almost all cells of your body. Calcium levels remain pretty low inside. So just kind of standard body physiology is almost always, we're gonna find, what happened to my marker? We're gonna find lots of calcium outside of all of our cells in the ground substance, or at least more than in the cytoplasm of the cells. So if there's a higher concentration of calcium outside, lower concentration inside, then calcium will diffuse into the cell in that direction. No energy is required, just the concentration gradient. Calcium is the trigger to cause exocytosis of these vesicles that contain neurotransmitter. So when calcium comes in, all these vesicles will exocytose their neurotransmitter. Acetylcholine spills into the synaptic cleft, diffuses across through that fluid, and can now be seen by the acetylcholine receptors down here in my myofiber. So this is going to be an ACH receptor. So when the receptor finds acetylcholine, that's the trigger for the myofiber to initiate contraction. So we've got half the story. We've got another half of the story to go. But a lot of what we saw up here does get replicated down here. set the stage for ourselves. But we need a break. It's a lot. Questions on this so far? Okay, I do want you, before I give you the break, I do want you to recognize that in teaching this stuff, right, there's a lot of physiology here. This is probably the most physiology we get into in anatomy. But the focus for us is the anatomy. The focus for us is where are these channel proteins located, right? And I've tried to, instead of just say, you know, we could get through this in 10 minutes if I just say, oh, ligand proteins are here, voltage-gated proteins are there. But now you can make sense of it, right? Hopefully, by understanding how they all work and understanding this process, maybe you can go back and make sense of, oh yeah, ligands have to be triggered by a neurotransmitter. That's why they're only found on the receiving end of a neuron. Voltage-gated, right, they're triggered by a change in charge. That's why they're only on the axon. Calcium plays a special role in exocytosis of a neurotransmitter. That's why it's only at the axon terminus. So that's my goal with explaining all of this, is to hopefully make it a little bit easier to remember where these things are located. Remember the anatomy. Take a break, we'll come back. I'm gonna go ahead and add in a few like sarcomeres. Remember the alternating myosin and actin? So I'm gonna build on my myofiber right now. Let's go 10 minutes, and then at like 9:52, we should be ready to go.
So what are we mixing? You want to know where the